A very good morning, friends. Today we will learn about an important topic in biochemistry, that is coenzyme. The term coenzyme, cofactor and prosthetic group, these terms are used loosely by various authors. These coenzymes, they are also known as co-substrate and secondary substrate, because these coenzymes, they also take part in the reaction. These coenzymes, these are non-protein small molecules or metal ions that participate directly in substrate binding and catalysis. They are known as co-substrate or secondary substrate. If we take one example, in that lactate, with the help of lactate dehydrogenase, it is converted into pyruvate. Along with this conversion of lactate into pyruvate or oxidation of lactate into pyruvate, there is simultaneous reduction of NAD into NADH and H ions. So this coenzyme also take part in the reaction. That's why it is known as co-substrate or secondary substrate. Now coming to these terminologies, the functional part of an enzyme, it is known as holoenzyme. Suppose this is a holoenzyme. It is a functional part of an enzyme. It is made up of protein part that is known as apoenzyme plus non-protein part. This is holoenzyme. It is composed of apoenzyme and coenzyme. This non-protein part of an enzyme, it is known as coenzyme. This non-protein part can be coenzyme, cofactor or prosthetic group. Now coming to the definition of coenzyme, cofactor and prosthetic group. In general, these are the terms used for the small molecules or small non-protein molecules and metal ions directly participate in an enzyme catalyzed reaction. Means these are the small non-protein molecules and metal ions associated with enzyme action. They are known as coenzyme, cofactor and prosthetic group. Now coming to the coenzyme, you can remember the definition of coenzyme by this mnemonic, that is, coenzymes are non-protein, organic, low molecular weight and dialyzable substance associated with enzyme action. Non-protein, organic, low molecular weight and dialyzable substance associated with enzyme action. It is easily separable. Dialyzable means separable or easily dissociates from the apoenzyme. They are known as coenzyme. Now coming to the prosthetic group there is tight and stable association between prosthetic group and apoenzyme. While in case of cofactor, there is weak and transient association with apoenzyme. So this is the operational differentiation or operational difference between these three terms. The coenzymes, they are organic molecule, they are dialysable, easily separable from the apoenzyme, they are non-protein. Then prosthetic group, usually they are metal ions in most of the cases. They are attached with tight and stable orientation or tight and stable association with the apoenzyme molecule. These enzyme molecules which contain prosthetic group, they are known as metalloenzymes. Example, zinc containing enzyme, that is carbonic anhydrase, or superoxide dismutase. Then xanthine oxidase contain molybdenum, glutathione peroxidase contains selenium. These are the examples of metalloenzyme. While in cofactor, these cofactors are in weak and transient association with the apoenzyme. These enzymes are known as metal activated enzyme. Example is ATPase. The metal ions are magnesium and calcium. So it is weakly associated with the apoenzyme or weakly associated with the ATPase. It can be easily separable. So this is the operational difference between these three terms: coenzyme, prosthetic group, and cofactor. Now coming to the classification, or you can say it examples of coenzyme. They are divided or classified on the basis of their precursor molecule. First, they are classified on the basis of their precursor molecule. There are two types: coenzymes derived from B complex and non-B complex coenzymes. Then second classification, those who are involved in the transfer of hydrogen ion transfer, or those who are involved in oxidation reduction reaction. And second is those who are responsible for transfer of group other than hydrogen ion, or transfer of other groups. And third example is metal ions. Metal ions which act as a cofactor. So these are the three examples of coenzyme. Now coming to the B complex. Start from the B1, B2, B3, B5, B6, B7, B9 and B12. These are the B complex vitamin. B1, it is thymine, and the coenzyme form of thymine is the thymine pyrophosphate. It is involved in the transfer of aldehyde or keto group. The example of enzyme is transketolase. It is in the HMP shunt. So this is the first coenzyme, that is TPP, thymine pyrophosphate. Second is the riboflavin. Coenzyme forms are FMN and FAD. Flavin mononucleotide and flavin adenine dinucleotide. It is involved in the oxidation reduction reaction, or it is involved in the transfer of hydrogen ions. The examples here, the FMN example is amino acid oxidase, FAD is succinate dehydrogenase. Amino acid oxidase, succinate dehydrogenase. B3, niacin, coenzyme form NAD, NADP. It is also involved in oxidation reduction reaction. Examples: lactate dehydrogenase, alcohol dehydrogenase, pyruvate dehydrogenase complex. Then B5, pantothenic acid, the coenzyme form is coenzyme A. It is involved in the transfer of acyl group. Example is pyruvate dehydrogenase complex. Then B6, that is pyridoxine. Coenzyme form is pyridoxal phosphate. It is involved in the transamination reaction. It is responsible for transfer of amino group. Example SGOT, SGPT, serum glutamate oxaloacetate transaminase, serum glutamate pyruvate transaminase. Then biotin, coenzyme form is biocytin. It is responsible for transfer of carbon dioxide, and it is important in carboxylation reactions. Many carboxylase, they require ABC. Carboxylase require ABC, ATP, biotin and carbon dioxide. Then folic acid, the coenzyme form is tetrahydrofolate. It is responsible for transfer of one carbon atom in various reactions. Then B12, the coenzyme form is adenosyl B12 or methyl B12. This adenosyl B12, it is involved in isomerization reaction, that is methyl malonyl coenzyme A mutase, and this is involved in methylation reaction, that is methionine synthase. So these are the different coenzymes of vitamin B complex. The non-B complex coenzyme, it includes, the mnemonic is campus, C-A-M is silent here, P-U-S. The examples are CDP, ATP, phosphoadenosine phosphosulfate, UDP, and S-adenosyl methionine. These are the examples of non-B complex coenzymes. CDP, it is involved in transfer of choline and ethanolamine. In the synthesis of phospholipid. ATP, it is responsible for transfer of phosphate, transfer of AMP, transfer of adenosine, etc. PAPS, it is an active sulfate donor in the synthesis of mucopolysaccharides. UDP, it is responsible for transport of glucose and galactose. It is main coenzyme, or it is important coenzyme in the glycogen synthesis. Then S-adenosylmethionine, it is important in transmethylation reaction. So these are the different examples. These are the B-complex coenzymes. These are non-B-complex coenzymes. Other classification includes those who are involved in oxidation reduction or transport of hydrogen ion. This includes this FMN, FAD, NAD, NADP and coenzyme Q. FMN, FAD, NAD, NADP and coenzyme Q. They are involved in the oxidation reduction reaction or transfer of hydrogen ion, and all others, they are involved in the transfer of group other than hydrogen ion. This is the second classification. And third example is some cofactors or some metal ions that acts as a cofactor. So this includes for example zinc. It is important in many, more than 300 enzymes that contain zinc. It is an integral part, metalloenzyme. These are carbonic anhydrase, superoxide dismutase, carboxypeptidase, then alcohol dehydrogenase and RNA polymerase. These are the important enzymes which contains zinc as an integral part. Then copper, it is present in cytochrome oxidase, phenol oxidase, tyrosinase, etc. Selenium, it is in glutathione peroxidase. Then molybdenum in xanthine oxidase. So these are the important cofactors or important minerals which act as a cofactor for many enzymes. So this is all about coenzyme. Summarizing all, coenzyme, cofactor and prosthetic group, they are differ only in their operation. For example, cofactors, they helps in catalysis when they are present surrounding the enzyme molecule. But prosthetic group, it is present in tight association with the apoenzyme molecule. There is only operational difference between these three. Then coenzyme, it is also known as co-substrate or secondary substrate because coenzyme also take part in a reaction. Then there are different classification based on precursor molecule and based on reaction they catalyzed, and some metal activated enzymes and metalloenzymes are there. So this is all about coenzyme. Thank you. Keep watching.